So basically, we are talking about the transport through the xylem and transport through the phloem, right? Here we will understand the things in a different way, right? You must have studied the same chapter in class 10th as well. So here we are going to discuss, like I'll tell you how we are going to discuss this thing. So in case if any one of you have any doubt regarding the previous classes in animal physiology and endocrine system, then please ask me if you have any doubt. Now the next will be transport. So, and the first topic will be means of transport. So now let's start with our topic, means of transport. So what are the means of transport? How does the transport takes place? Let me check the NCERT as well. We follow the same sequence of the NCERT. So means of transport. So first, when we talk about the transport, what we are talking about, that's what I want to tell you. I want to give you a brief about the means of transport. So what are the means of transport? How does the transport takes place in plant, right? So when we talk about the means of transport, we are talking about, look, in means of transport, there are two types of the transport. Short distance and long distance transport. So short distance and long distance transport. So when we are talking about the short distance transport, then how does the transport takes place? While you are talking about the short distance transport, then we are talking about the transport of material from one cell to another cell. So in this way, the transport takes place. How does the transport takes place? So whenever we talk about the transport for the short distance, then we are talking about the cell to cell transport. So for sake of example, suppose that these are the two cells and transport from one cell is taking place to another cell. So how does the transport will take place? In these cells, the transport will take place from one cell to two cell. There may be one process that may be diffusion, right? It may be due to diffusion. Diffusion, or it may be due to osmosis. So these are basically the process by which the transport takes place in short distance from one cell to another cell, right? We'll discuss all these things in detail, right? And these may be due to even it may be active transport as well. It may be active transport. So that may be active. We can't say the diffusion. You get the active transport. How does the transport takes place for the long distance? When we are talking about the long distance transport, it means the transport is taking through with the xylem. So xylem is meant for the transporting water plus mineral. Water plus mineral. Then it's phloem. Phloem is responsible for transport of organic substances. Organic substances. Right. So it transport organic substances. So these are the two ways of transport, right? Now, what are the means of diffuse transport? So means of transport, if either it's long distance or it's short distance, the transport will take place either through the diffusion or through the osmosis or through the active transport, right? So these are the means of transport. So that's how the transportation takes place. Why the long distance transport is required? The long distance transport is required. Look, I am just giving you snapshot, nothing else. You have to just focus on and try to understand what we are studying. So when we talk about the long distance transport, for sake of example, suppose that this is a tree. This is a tree. This is a tree, right? In this tree, for sake of example, this is a leaf. This is a leaf. So now what happened? There are certain kind of the tubes, certain kind of pipes. These are the xylem. They send it because they cannot leaves directly cannot take the water and mineral. So these are the pipelines which transport their xylem. So what they do, they actually transport the water and mineral. In return, what root require? Root cannot do the photosynthesis because they are not green. They do not have chlorophyll like of the substance. In return, leaves give them back the organic substance, and that is the food. So these are the phloem. So phloem are responsible for providing food, right? So that's how the long distance transport takes place with the help of xylem and phloem, right? Now let's start with the uh, means of transport. What are the means of transport? Means how does the transport takes place? What are the processes which help in the transportation, right? If you want to note it down, please keep on noting down, right? Keep on noting. This is the just snapshot. I want to give you the snapshot. How does the transport takes place? Why transport takes place? So look, when we talk about the means of transport, what are the means which help in the transport? So look, in transport, basically there are two type of the transport. First, it is active, and then this is passive. The difference between the active and passive transport is that, look, in general, first we talk about the passive transport. In general, in passive transport, the transport takes place with the help of diffusion. Diffusion means that is molecules always moves directly from low concentration, low concentration to its higher concentration. So when I say this is the movement of molecule is taking place from low concentration to higher concentration, you directly call it diffusion, right? So, but I say this is the diffusion is taking place or transfer is taking place in the direction of diffusion gradient, the direction of diffusion gradient. So this is called diffusion gradient. So when transport takes place from low concentration to higher concentration, we call it diffusion gradient. In diffusion gradient, so the passive process here, no ATP is required. In this process, no ATP, that is no energy is required. No energy is required, right? Here energy is not required. So this is called the means of passive transport. In passive transport may be of following type, right? It may be of two types. First, this is simple diffusion. So there are two things for the passive transport. Energy is not required for that. It will always be in direction of diffusion gradient, right? So this may be simple diffusion. And the second process is called facilitated diffusion. Facilitated diffusion. Facilitated diffusion. So diffusion may be two types: simple diffusion and facilitated diffusion, right? And further facilitated diffusion also have a further category. We'll discuss for that. But when we talk about the active transport, so what is the active transport? In active transport, ATP is required. First thing, ATP is required. Second, first thing, ATP is required. And what is the second thing? Second important thing is that It takes place. It takes place against the against concentration gradient. Concentration gradient. What is that? That is, it takes place from higher to low concentration. Higher to low concentration. Low concentration. This we call uphill concentration. Like lower to when the diffusion is taking place, or when the transport is taking place against the concentration gradient, we call it uphill. When diffusion takes place towards the direction of concentration gradient, we call it downhill. Right? Please note it down. Note it down. Then I will start. When you note it down, please let me know. Done?
Yes, sir.
Right. Please note it down. Substances move over the short distance. Substances move over the short distance through diffusion and active transport. Substances move over the short distance through diffusion and active transport. Through diffusion and active transport. Supplemented by cytoplasmic streaming. Supplemented by cytoplasmic streaming. Full stop. Next point.
So cytoplasmic?
Cytoplasmic streaming. Cytoplasmic streaming. Full stop. Long distance transport. Long distance transport occurs through vascular system. Long distance transport occurs through vascular system. Vascular system, comma, xylem and phloem. Vascular system, xylem and phloem. Full stop. It's very important thing. The next point, what I am going to dictate, you please make a star for this. The direction of translocation. The direction of translocation. This is question, it have been asked. Direction of translocation is unidirectional. Is unidirectional in case of water. Direction of translocation is unidirectional in case of water. That is from root to stem, leaves, flowers and fruit. Root to the stem, leaves, flower and fruit. Full stop. The translocation is essentially multidirectional. This was the question actually. Translocation is essentially multidirectional in case of phloem. In case of phloem, where mineral and organic solutes, are translocated. Right? Done. So this was now you have to put the heading means of transport. Right? So now I am talking about the means of transport. Look. So first I told you active and passive transport. You have noted down it. Now I am talking about the. I'll show you a diagram. Look, passive transport and active transport. Active transport takes place at the expense of energy, right? And when we talk about the passive transport, so in passive transport, so this is the plasma membrane. This is the plasma membrane where in diagram you can see the bilayer of lipid, right? Look, this is the bilayer of lipid, right? Lipid, and this is the plasma membrane, and these are the actually intrinsic protein. These intrinsic protein, what they actually do. So look, whenever we talk about the there are certain substances, those substances which are lipid soluble. So lipid soluble substances, they directly cross through this bilayer of lipid, right? So when I talk about the lipid soluble substances, means we are talking about the hydrophobic substances. So hydrophobic or little lipid soluble substances, they directly diffuse from the higher concentration to lower concentration. So lipid soluble substances, they cross the plasma membrane with the help of diffusion. Please try to understand. When I talk about those substances which are water soluble, right? Water soluble. So when I am talking about the water soluble, means I am talking about the hydrophilic substances. Right? Hydrophilic. So water soluble or hydrophilic substances, they cannot cross through the plasma membrane directly through the lipid bilayer. They have to pass through the protein. These protein which are facilitating or which are providing the facility to pass these molecules, so these protein, these are intrinsic protein. Basically these proteins are called, they are basically called channel protein, right? So they are basically called channel protein, right? We call them channel, or in case of iron we also call them pump. Is it clear or not? They are called channel protein or pump protein. Look, now I am telling you an example, right? So when I talk about the channel proteins are basically of two types, right? They may be simply pump, or there may be a carrier protein as well. What is the function of carrier protein? How does it work? Look, suppose that you are in your school, and in your school there is a bus, right? A school bus. Now this is the time to leave the school, right? And this is the time of you're signing off from the school. So what happens? Bus is in front of your classroom or in front of your school. So what will happen? There is no students inside the bus, and out of the bus there are number of the students. So student will diffuse from the higher concentration to low concentration. You people study in class 12th, you are grown up children. So what will happen, you will go by yourself, and you all these people, they move out or diffuse without any expenditure of energy, right? They go directly inside the bus, right? So the movement is taking place from higher concentration of student to lower concentration of student, and the gate in the bus, you call that what? Channel or pump. So you are passing through the channel and pump. This was the one scenario. So this is the passive transport, and passive transport with the help of channel. When I talk about the carrier protein, what's the carrier protein? In the same time, a bus is aside you, and that bus is meant for, and it is providing facility for those students who are from in class 1 standard or play group or kinder group, right? So those nursery students, they cannot ride in the bus by themselves because their height is so short. So what will happen? School will provide a facilitator, right? There will be a male or female who will put the hand of a small kid, and it will like pull him or her into the bus, and it is helping them to boarding on, right? That person, this is also a passive transport, that particular person, so that gate will be called carrier protein, right? So that gate will be called channel protein, and inside the person who is pulling these kids, small kids, that is called carrier protein, right? Is it clear or not?
Yes, okay.
Yes, sir.
So in facilitated diffusion, it maybe takes through the channel protein, and even it may be takes place through the carrier protein, right? Carrier protein. So these are the, and when we talk about the active transport, it takes place due to expenditure of energy, right? You can take the screenshot of this one. And now there may be type of carrier protein, right? Carrier protein may be of following type, and we will study the carrier protein. First, please take the screenshot and start noting down. The first, diffusion. Diffusion is movement through passive process. Diffusion is movement through passive process, and it's slow. Diffusion is movement through passive process, and it's slow. Full stop. It occurs along concentration gradient. It occurs along concentration gradient. That is, it occurs along concentration gradient. That is, from higher to lower concentration. That is, from higher to lower concentration. Full stop. Expenditure of energy do not takes place. Expenditure of energy do not takes place in this process. Expenditure of energy do not takes place in this process. Full stop. Next point, facilitated diffusion. Second heading, B, facilitated diffusion. Particles which are not lipid soluble, particles which are not lipid soluble, that is hydrophilic solute, that is hydrophilic solute, cannot pass through lipid membrane. Full stop. For this, the membrane possess a particular type of protein, called channel. Full stop. Channels, right? There are 8 different kind of channel. There are 8 different kind of channel which are known as, so these are called channel or ion channel or pump, right? So these are, rather than pump, we call it ion channel, right? Ion channel. So these are also known as, these channels are also known as aquaporin, right? Because they carry hydrophilic substances. So this question have been asked in medical examination. So how many type of aquaporin are for? This is also in your NCERT as well, right? So these channel are called aquaporin, right? And the type, there are 8 different type of aquaporin are found. That question have been asked in medical examination twice, right? So please note it down. The 8 different type of aquaporin or water channel, 8 different type of aquaporin or water channels have been recorded. Full stop. Aquaporins are protein which transport, aquaporin are protein which transport water soluble substances passively. Water soluble substances passively. Full stop. Energy is not required. Energy is not required in this transport, as energy is not required in this transport as it is taking place in the direction of concentration gradient. It is taking place in the direction of concentration gradient. Full stop. Now carrier protein, right? There are 2 major type of transport protein. There are 2 major type of transport protein called carrier protein and channel protein. There are 2 major type of transport protein, carrier protein and channel protein. Carrier protein and channel protein. Full stop. Carrier protein bind the particular solute to be transported carrier protein bind a particular solute to be transported and deliver same the other side of membrane, and deliver same to other side of membrane. Full stop. Channel protein allow diffusion of substances of appropriate size. Fine. Is it clear or not? Please tell me. Channel protein and carrier protein difference clear?
Yes, sir.
Great. Now the carrier proteins may be of following type, right? Next point. The carrier protein may be of following type. Let me show you the type of carrier protein. carrier protein. It may be of following type, right? Look, carrier protein may be of following type, right? So there are two types. When they transport two substances simultaneously, they are called co-transport. And when they transport only single substance, single molecule, they are called uniport. So in uniport, they are transporting only one type of substance. In co-transport, they are transferring two substances simultaneously, not single, two substances simultaneously. For sake of example, suppose that if they are transporting for potassium, right? So when we are talking about when they are transporting potassium, this potassium transport, that is the uniport. And you must have we already have studied the sodium potassium pump in nervous system. So in sodium potassium pump, we have studied when the sodium goes inside, then potassium comes outside, right? So they are called antipode. They transport simultaneously but in opposite direction. They are called antipode. When we called about a sympode, I'm also giving you one example, sympode. So antipode sodium potassium pump we have studied. This is the sodium potassium pump. We have already studied in the case of nervous system sodium potassium pump, right? And when we talk about the sympode we have studied in digestive system. This question also have been asked in medical examination. So if sodium also help in transport of what uh, that is uh, fructose. The transport of fructose, sodium and fructose are transported simultaneously. They are example of sympode. So sympode, they transport two substances in same direction, right? Two molecules in same direction. Antipode, they transport two substances, but the direction will be opposite. In unipode, they will transport only one substance. To carrier protein may be of following type. Clear? Sympode, antipode, and unipode. Clear? Minashi, clear? Samaira,
Yes.
clear?
Yes, sir.
Great. Please note it down. Take the screenshot and please note it down. Please draw the diagram. Always draw the diagram. Please note it down. Co-transport. Some carrier protein allow transport. Some carrier protein allow transport. Some carrier protein allow transport only if two type of molecule moves together. Only if two type of molecule moves together. Full stop. This type of transport, this type of transport called co-transport. This type of transport is called co-transport. Full stop. It may be of two type. It may be of two type. Full stop. In sympode method of co-transport, in sympode method of co-transport, in sympode method of co-transport, both molecule cross membrane. In sympode method of co-transport, in sympode method of co-transport, both molecule cross the membrane in same direction. Both molecule cross the membrane in same direction at same time. Full stop. In antipode method, both molecule moves in opposite direction. In antipode method, both molecule move in opposite direction. Full stop. Next point, unipode. When a molecule move across membrane, independent of other molecule. The process is called independent of other molecule. The process is called unipode. The process is called unipode. Right? Next is, so this was all about the passive transport. Let me know, is it clear or not? Please tell me. Is it clear?
Yes, sir.
100% clear? This is very important part. This is the very important part of this one. This thing, and the second one is that is the water potential and all that concept. These two things are very important, right? So if you says it's yes, sir, then I believe on you. So please, next heading is, so we talk about the passive transport, and we studied passive transport diffusion, then we studied facilitated diffusion, then we studied channel, then we studied carrier, then we studied the types of carrier protein. Next, put the heading active transport. In active transport, the movable carrier protein are called pump. In active transport, the movable carrier protein are called pump. Full stop. They employ ATP energy. They employ ATP energy for transport across membrane. They employ ATP energy for transport across membrane. Full stop. It is uphill transport. That is, it is uphill transport. That is, it takes place. It is uphill transport. That is, it takes place along the it takes place against the concentration gradient. It takes place against the concentration gradient. Full stop. It is faster than passive transport. It is faster than passive transport. Full stop. Carrier proteins are highly specific like enzymes. Carrier protein is highly specific like enzyme. Yes, guys, tell me, is it clear? Uniport, symport, antiport, and all that. 100% clear to all of you?
Can you repeat?
Carrier proteins are highly specific like enzyme. Carrier proteins are highly specific like enzymes. Clear to all of you?
Sir.
Right, great. Now there is a small topic, and that is called imbibition. Do you know what is that, imbibition? Any one of you have any idea?
Inhibition?
Imbibition. Let me write it down.
So is it like the lock and key method, like the enzymes will stop other enzymes from binding, something like that?
Ambivision.
No, like inhibitors will stop other enzymes from binding.
Anyone else? Ambivision. That is inhibition. No, this is imbibition. You are talking about the inhibition. That is inhibition.
So not
This
sure.
is this is imbibition. IMBIBITIUN. Imbibition. Look, simple. Suppose that I have a wood. I just poured this wood inside the any just leave the that wood. Look, I have ice cream and I have a melted chocolate. I put the ice cream. I like poured inside the melted chocolate and it pulled it out. Then what will happen? The outer surface of the ice cream that will absorb the chocolate, but will it go inside the ice cream? No. So whenever any solid surface absorb water or some other substances, and it takes place basically in outer surface, that is called imbibition. For example, in the rainy season, the door and windows of making made up of woods, they become tight, and soaking of water by the seed, right? That is also called imbibition. Clear? So please note it down. The absorption of water by solid particle, absorption of water by solid particle of an absorbent, the absorption of water by solid particle of an absorbent causing increase in volume, causing increase in volume without forming solution, causing increase in volume without forming solution. It is called imbibition. Without forming solution, it is called imbibition. Example: seeds, dry wood, they absorb water. Right. Now the next topic is that plant water relationship. So when I talk about the osmosis, plasmolysis, and we'll start with it. So now we will talk about the next topic. For the next topic, plant water relationship. Diffusion pressure, and BPD. Now let's talk about the osmosis, right? First, I will teach you osmosis. Then I will teach you what is the diffusion, then diffusion pressure, what then diffusion pressure deficit, right? DPD. So first, talk about the osmosis. We'll study gradually, one by one. So, what is osmosis? Yes, this is. You already know what is the osmosis process, but still we have to study that. Look, there are two solutions, right? So there are two solutions. So out of them, sorry, out of them, this is pure water, right? This is pure water. So it's have pure, like lots of water molecule. Here, this is sugar solution, right? This is solution, right? So this is solution. And we have separated both of them with the help of a differently permeable membrane. This differently permeable membrane is also known as semi-permeable membrane. Same thing. Semi-permeable membrane. So whenever we talk about the differently permeable or semi-permeable membrane, they allow the movement of only water molecule, not sugar molecule. Only the water molecule can pass through them, right? So water molecule will start moving from its higher concentration to lower concentration. So whenever we talk about the movement of, so what is the osmosis? Osmosis is the process of movement of water molecule from its higher concentration to lower concentration. Water molecule, or we can say osmosis is the movement of water molecule from low concentrated solution towards higher concentrated solution, or we can say its take part. So low concentration is also called movement of water molecule from hypotonic solution to hypertonic solution. Hypertonic solution, or we can say movement of water molecule from its higher concentration. Now we are talking about the individually water molecule. So water molecule concentration is higher over here and lower here, right? Movements of water molecule from its higher concentration to lower concentration. All these things are same. Is it clear or not? Tell me. Clear? Osmosis clear?
Yes, sir.
Yes, sir.
Please take this screenshot and note down these three things. These three things, whatever I have noted down, that hypo to hyper.
Done, sir.
Done? So please note it down. Diffusion of water molecule from its pure state or dilute solution. Diffusion of water molecule from its pure state or dilute solution into a solution or stronger solution. Diffusion of water molecule from its pure state or dilute solution into a solution or a stronger solution when separated by semi-permeable membrane. When separated by semi-permeable membrane, 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 is termed as osmosis. Next point, osmosis is movement of osmosis is movement of solvent or water molecule. Osmosis is movement of solvent or water molecule, from the region of their higher diffusion pressure to lower diffusion pressure, from higher diffusion pressure to lower diffusion pressure. Is it clear?
So can you explain diffusion pressure?
Diffusion pressure, look, these molecule have low, like higher free energy, right? And these molecule have lower free energy. Why? Because these molecule do not have any sugar solution. Here is sugar solution is available, so these molecule will collide with sugar solution, hence their energy decreases, right? So they have less free energy, they have high free energy, right? So these high free energy molecule will cross this membrane, and whenever they cross this membrane, they impart a pressure, right? This red arrow is showing the pressure. They impart a pressure, and this pressure is called diffusion pressure. Clear? So diffusion pressure we can determine that from higher energy to its lower energy. Clear?
Yes, sir.
Okay. The next thing is that I want to tell you what is the diffusion and diffusion pressure. Again, I'll define the same thing: diffusion and diffusion pressure. So next heading is diffusion and diffusion pressure. That is called DP, right? So what is diffusion? Diffusion we know. Please note it down. What is diffusion? So diffusion is tendency of molecule. Diffusion is tendency of molecule, atom or ion, of gas, liquid or solid. Tendency of molecule, atom or ion, etc., of gases, liquid and solid, to get evenly distributed throughout the available space, on account of random kinetic energy. Is called diffusion. So that is diffusion. So for sake of example, they have like the same thing as we said. So suppose that there is a separation and there is a wall between them, this separation, and here these are the water molecule, and here there are some water molecule are less over here, and here along with the water molecule there are some salt molecule. So what will happen? The movement of water molecule will take place from its higher concentration to low concentration. We call that diffusion, right? So these water molecule would like to spread evenly, so that even distribution of water molecule will take place. That is called diffusion, right? That is simple called diffusion, right? What is diffusion pressure? So please note it down. Diffusion pressure: the pressure exerted by particle, the pressure exerted by particle to diffuse from area of the pressure exerted by particle to diffuse from area of higher concentration to lower concentration is called diffusion pressure. Full stop. No, that is a diffusion pressure, right? So we call it dP. Now we have studied osmosis, diffusion, then yes, one more important thing, diffusion pressure deficit. One more important which I want to tell you. So this was diffusion pressure, and one more thing that is called dPd. dPd. dPd is also called diffusion pressure deficit, right? Now, what is diffusion pressure deficit, right? So, suppose that, for sake of example, the diffusion pressure of molecule present over here. This is the A part, and this is the D tumbler, right? Or we can say B tumbler, right? So here, the diffusion, for example, diffusion pressure is 5 mmHg. That is diffusion pressure. Here, the diffusion pressure is 1 mmHg. So the diffusion will take place until it becomes equal at both of that. So diffusion will take place until 3 becomes here and 3 becomes here. Now the deficiency of here is 2. So there is a 2. So 2, the pressure, what pressure is deficient over here? The pressure which is deficient over it, that is a 2. So that is 2 will be what? dPd, diffusion pressure deficit. Is it clear or not?
So I can explain once more.
One look. In this A tumbler, try to understand me. In this A tumbler, there is 5 mmHg diffusion pressure. In B tumbler, there is 1 mmHg diffusion pressure. Out. So then the molecule will start diffusing out from their higher concentration to low concentration. When then diffusion will stop? When the process will stop? The process will stop when the diffusion pressure become equally at both of them, right? Clear? So when at both of the end, pressure will become equal, like 3 mmHg here and 3 mmHg here. Then we will call that this is the equilibrium state. This is called equilibrium state. And at this state, equilibrium state, the diffusion will stop, right? So now the 1 mmHg must become to 3 mmHg. So what is, how much deficient over here? Deficiency is of 2 mmHg, right?
Yes, sir.
So that deficiency will be called diffusion pressure deficit. The deficit means the less in diffusion pressure. So diffusion pressure is 1 mmHg. What should be? It should be 3 mmHg, right? So 2 mmHg is left. That is the diffusion pressure deficit. Clear or not? Please tell me. Minashi, Samira, clear?
Yes.
Yes, sir.
Okay. So please note it down. Alvia, clear?
Yes, sir.
Okay, great. So please note it down. Diffusion pressure deficit. Pure water have maximum diffusion pressure. Obviously, pure water have maximum diffusion pressure. Full stop. Pure water have maximum diffusion pressure. Full stop. Its diffusion pressure gets lowered by addition of solute particle. Its diffusion pressure gets lowered by addition of solute particle. Full stop. The reduction of diffusion pressure of water, the reduction of diffusion pressure of water in a solution, the reduction of diffusion pressure of water in a solution over its pure state, the reduction of diffusion pressure of water in a solution over its pure state is called diffusion pressure deficit. Is called diffusion pressure deficit. Clear? Now I want to tell you some more term. What is the wall pressure? What is the turgor pressure? Right. So now it will become more clear. Look. Please try to understand this diagram. This is the perfect diagram to explain all these concepts. Suppose that a cell is kept in water. So what will happen? Water will start coming from outside to inside the vacuole of cell. So osmosis will take place. Clear? So whenever the water will move from its higher concentration to lower concentration, they will impart a pressure, and that is called osmotic pressure. Clear? This
Yes, sir.
molecule will move from higher to lower concentration, right? Higher concentration of water to lower concentration of water. They will impart a pressure on that plasma membrane or a membrane. That is called osmotic pressure. Clear? So direction of the osmotic pressure inward, right? Now the water will start accumulating in vacuole. Now it will start expanding the cell. It will start expanding outward the cell. So it will drop a pressure to outside in the direction of outside. That is called turgor pressure. So now the cell will swell, and swell cell or swelling. Swell cell is called turgid cell. Turgid cell. So when cell will become turgid, it will this water will drop pressure outside. That is called turgor pressure, TP. Now what will happen when turgor pressure TP will push this cell outward? Then the cell wall will try to maintain its shape, and the cell wall of the particular cell will put the pressure or drop the pressure in inward direction. That is called wall pressure. Is it clear or not clear?
Yes.
OP, TP, and WP. Clear?
Yes, sir.
Fine. So there is a formula for DPD. The formula in DPD is that DPD. So one more thing I want to tell you. this turgor pressure and wall pressure. This is in opposite direction and they will always be equal. So TP will always be equal to WP, wall pressure. So turgor pressure will always be equal to wall pressure. Keep it in mind, right? Always. Now, what is the formula for DPD? The formula for DPD is equal to osmotic pressure minus wall pressure, or wall pressure is equal to turgor pressure. So you can also say osmotic pressure is minus TP. Both are the same. That's the DPD. Clear?
Yes, sir.
Yes,
Please
sir.
note it down and take the screenshot. This is very important. Note it down and let me know. Done?
Yes, sir.
Okay. Now look, we have studied about the osmosis. That was that in osmosis. Let me again recap it. Osmosis, what we studied in osmosis. So this osmosis. We have studied that movement of water molecule takes place from pure water state to solution state, from low concentration of solution to higher concentration of solution, right? From hypotonic to hypertonic, right? That's it. So these are the older concepts. Nowadays, we are talking about the water potential. Now, what is the water potential, right? So now the concept is of water potential. Water potential. Water potential is a term. We represent this potential term or this water potential in the term of with a letter, and that letter is called psi. This potential term, this is called psi. And when we talk about the water potential, so psi W means water potential. What is water potential? Water potential is the free energy of water molecule, right? So when we talk about the free energy of water molecule, it is maximum at its pure state. So it have maximum free energy, right? Maximum free energy. Whenever we mix any solute with water, so it becomes a solution. In solution state, the energy of water molecule, free energy of water molecule reduces, right? So this is called like in solute state, this is low free energy of water molecule. So water molecule moves from its higher free energy towards the lower free energy states through a semi-permeable membrane. This is called what? Water potential, right? So water potential is maximum water potential of pure water, right? So psi W of pure water, psi W of pure water, what? So this is always 0. So maximum water potential will be always 0 because the water potential of pure water is 0, right? So whenever we will mix any solute with the pure water, then what will be the water potential? It will always be what? Negative, right? Is it clear or not clear? Water potential of pure water is 0. So in solution state, what will happen? It will always be negative, right or not?
Yes, sir.
Yes.
Okay,
Can you
fine.
repeat?
Right. So water potential of solution will always be negative. So water molecule moves always from its higher free energy to lower free energy. That process called water molecule. One more thing. There is another term that is called psi s. Psi s is called solute potential. So what is solute potential? Psi s. This is called solute potential. Solute potential is the amount of water energy or water potential that reduces due to mixing of solute, right? There is one more term, psi p. Psi p is called pressure potential. Pressure potential. What is that? That is the impact of pressure on water free energy of water, or impact of pressure on water potential. Solute potential, impact of solute on water potential. Pressure potential, impact of pressure on water potential. Is it clear or not? Same thing is osmosis. We are representing the osmosis in terms of water potential, right? Osmosis is the process that we explained in the terms of chemistry. Water potential is the process we explain in the terms of physics. That's it. Is it clear or not now? Please tell me. Yes, guys. Minashi, Samaira, Alvia, Atif.
Yes, sir.
Right. So please take the screenshot and note it down. Put the heading water potential. Chemical potential of pure water. Chemical potential of pure water at normal temperature and pressure is 0. Chemical potential of pure water at normal temperature and pressure is 0. Let's stop. In solution, the value of water potential is always negative. In solution, the value of water potential is always negative or less than 0, or less than 0. Full stop. It is represented by Greek symbol psi. It is represented by Greek symbol psi. Full stop. The value, so what is the unit of water potential, right? So the value of water potential is measured in bars, Pascal, or atmosphere. The value of water potential is measured in bar, Pascal, or atmosphere. Full stop. The movement of a substance, from area of higher free energy, movement of substance from area of higher free energy to lower free energy, the movement of substance from area of higher free energy to lower free energy is called diffusion. So what is the formula for psi W? So there is a relationship between these all terms. So the relationship is that psi W is equal to psi P plus psi S. So that is the water potential is equal to solute potential pressure potential. Noted on the formula. Next point. Solute potential is decrease in the chemical potential of pure water. Solute potential is the decrease in the chemical potential of water, chemical potential of pure water, chemical potential of pure water due to presence of solute particle in it. Let's stop. Solute particle reduces the free energy of water. Full stop. Pressure potential is the effect of pressure. The pressure potential is the effect of pressure on the free energy of water molecule. Is that clear? Tell me, guys. Solute potential, water potential, pressure potential. Is it clear or not? All these terms.
Yes, sir.
So osmosis always takes place from higher water potential to lower potential. Diffusion always takes place from higher water potential to lower water potential. That is from 0 towards the negative. Clear? Okay. So
Yes, sir.
the next topic is plasmolysis. Right. So I will tell you, I elaborate term of plasmolysis. What is plasmolysis? I will tell you this process in detail. What is plasmolysis? What is the incipient plasmolysis? What is limiting plasmolysis? So put the heading plasmolysis. These are the types of plasmolysis. These are the actually stages of plasmolysis, right? So what will happen? This is the normal cell, and actually this is the plasmolyzed cell, right? So first stage of the plasmolysis, that is basically called limiting plasmolysis. This step is limiting plasmolysis. limiting plasmolysis, right? So this is limiting plasmolysis. What happened whenever a cell is put in hypertonic solution? What happened? Water coming out from the cell, right? Now, whenever the water will start coming out of the cell will become flaccid, right? The cell will become flaccid. Cell will become flaccid, means loose. Now, this is called limiting plasmolysis, right? Then cell will shrink and reduces its shapes. What will happen when further more hypertonic solution, the concentration of outer side solution increases? So now the more water come out from the protoplasm and vacuole. What happened then? The protoplasm will start leaving cell wall at corner, this space. Protoplasm start leaving corner space creation, right? So protoplasm will start leaving from the leaving cell wall at the corner. This stage is called incipient plasmolysis. When further more hypertonic solution is kept, what will happen? The protoplasm will leave the cell wall except 2 or 3 places. It's attached over here, it's attached over here, it's attached over here, and it have leave the rest of the part. And the space between the cell wall and plasma membrane, there will be hypertonic solution will go inside this space, right? So this stage is called evident plasmolysis. In final plasmolysis, what will happen? This protoplasm will shrink and this vacuole become minimized, and this protoplasm will completely leave the cell wall, and between this space all hypertonic solution will gush into and get accumulated, and this stage is called final plasmolysis. Is it clear or not? All stages clear?
Yes.
So please note it down. First is plasmolysis. First heading is plasmolysis. Shrinkage of protoplasm of a cell from its cell wall. Shrinkage of protoplasm of a cell from its cell wall under the influence of hypertonic solution. Under the influence of hypertonic solution is called plasmolysis. Under the influence of hypertonic solution is called plasmolysis. Full stop. Hypertonic solution causes exosmosis. Hypertonic solution causes exosmosis or withdrawal of water. Hypertonic solution causes exosmosis or withdrawal of water from cytoplasm, then central vacuole of cell. From cytoplasm, then central vacuole of cell. Full stop. Next, keeping the cell. Next stage, like no, first stage, that is a limiting plasmolysis. Keeping the cell in hypertonic solution causes reduction in cell size, due to exosmosis. Full stop. This is first stage. This is first stage of plasmolysis. This is first stage of plasmolysis called limiting plasmolysis. This is first stage of plasmolysis called limiting plasmolysis. It is first stage of plasmolysis called limiting plasmolysis. Next point. One more point in the same. This is called flaccid cell. This is called flaccid cell. Next, incipient plasmolysis.
Limiting plasmolysis.
Limiting plasmolysis, full stop. This is called flaccid cell. Next, incipient. Initially, the keeping shell in extra hypertonic solution, keeping shell in extra hypertonic solution, protoplasm withdraw itself, protoplasm withdraw itself from corner, protoplasm withdraw itself from corner, withdraw itself from corner. Let's stop. This stage is known as incipient plasmolysis. This stage is known as incipient plasmolysis. Full stop. Next, evident plasmolysis. Evident plasmolysis. The hypertonic solution now enter in the cell. The hypertonic solution now enter in the cell in between protoplast and cell wall. Now the hypertonic solution enter in the cell between protoplast and cell wall. Let's stop. Due to continued exosmosis, protoplast shrink. Due to continued exosmosis, protoplast shrink, and further withdrawal of cell wall, further withdrawal from cell wall, and further withdrawal from cell wall, except one or few point, it is known as evident plasmolysis. It is known as evident plasmolysis. Full stop. Next point. When the protoplasm completely leave cell wall, and when protoplasm completely leave cell wall and vacuole minimize its shape, this is called final plasmolysis. This is called final plasmolysis, right? One more point. There is a opposite process. What is deplasmolysis, right? So put the next heading, deplasmolysis. Deplasmolysis, suppose that they finally deplasmolysis. Plasmolysis. So in deplasmolysis, what happened? For sake of example, suppose that you have plasmolysed the cell, and again you put the same cell in pure water or in hypotonic solution. What will happen? if immediately after the plasmolysis you keep the cell in water or in hypotonic solution, cell will regain its original shape. This is called deplasmolysis. But this is only possible if you immediately keep the cell in hypotonic solution or in water, right? If you keep it after some time, then the reverse process will not be possible, right? Put the heading. And when the cell takes this water, this is called endosmosis. When cell remove the water or water goes out, this is called exosmosis, right? So deplasmolysis is the process of endosmosis, right? So please note it down. Swelling up of plasmolyzed protoplast, swelling up of plasmolyzed protoplast under the influence of plasmolyzed protoplast under the influence of hypotonic solution, under the influence of hypotonic solution or water is called deplasmolysis. Is called deplasmolysis. Full stop. And same point, deplasmolysis is possible. Deplasmolysis is possible only immediately after plasmolysis. Deplasmolysis is possible only immediately after plasmolysis. Now the next thing is that is the long distance water transport, right? Long distance water transport. So we have studied what is the long distance water transport. So next heading is water absorbing system, right? Under the long distance transport, we have a subheading, water absorbing system. How does the water is absorbed? Water water absorbing system. There is a two pathway, apoplastic or symplastic pathway, right? That help. Look, first let me explain you what is apoplastic and symplastic pathway. So water absorption takes place. So whenever we study the root anatomy, then outermost cell of the root anatomy, whenever we study about the root anatomy, then let me tell you, I want to show you one more diagram. So that will tell you the root anatomy. Even we have already understood that we have discussed the root anatomy, plant tissue part. So you must be remember with that. I want to refresh the same concept once again. Look, so we have already understood that the outermost part of the root that is made up of root hair and this part is called epidermis. So these are the epidermal cell and the elongation of epidermal cell is also called root hair. So these are root hair. So root hair are only just outgrowth of epidermis. The root hair, then epidermis, and between the vascular bundle and this part is called cortex. Cortex, the last layer of the cortex is endodermis. So endodermis is the last layer, right? Inside endodermis, there is a stele or this is called vascular cylinder. And when we talk about the vascular cylinder, we talk about the xylem and phloem. So these are the xylem vessels, right? So this is the cortex part. This is the epidermis. So outermost part is look. Now the outermost cells are of epidermis. Then it is the cortex. Then it is the inner part of the cortex that is endodermis. Then there is another layer that is called pericycle. Look, this is the endodermis, and inside the endodermis there is a pericycle, and inside the pericycle there is a xylem tissue, right? So now the movement of water takes place in between the two places. Look, first the movement of water molecule takes place between the space between the plasma membrane and cell wall. So this is the space between the plasma membrane and cell wall. Here the minimum resistance takes place. So water just move or translocate between the plasma membrane and cell wall. So this is the this place, right? So this is the between the plasma membrane and cell wall, right? So this is called apoplastic pathway, right? The first thing, apoplastic pathway. Again, when it enters to endodermis have a strip called Casparian strip. Casparian strip is impermeable to water. That is impermeable to water. That is impermeable to water. Hence, the water cannot cross to endodermis, right? So what happened? There are now here the apoplastic pathway is not possible. Then water enter inside the protoplasm, and it now transport through the protoplasm. This is called symplast, and with the help of plasmodesmata, again to the protoplasm, then again to symplast, right? So whenever we talk about these cells, right, there is another pathway of the water. In this pathway of the water, this is called symplastic pathway. In symplastic pathway, water moves through the protoplasm. So movement of the water takes place through the protoplasm only, right? So this is called symplastic pathway. So in symplastic pathway, the movement of water takes place only through protoplasm. This is slow, and here the apoplasm, the movement of water takes place between the space in cell wall and plasma membrane. So this is called symplastic and apoplastic pathway. Clear? Yes, guys. Clear symplastic and apoplastic pathway. Take the screenshot of this one, and the same diagram must be in your NCERT, and your school authority will definitely ask you this one. Even they will ask you what is the facilitated diffusion, what is the passive diffusion, what is the active diffusion. This is very important for your school examination. Active diffusion process, passive diffusion process, and facilitated diffusion, and all that, right? They will ask you. And the next diagram, they can ask you only the diagram in the 3 marks question. This one. Is it clear or not? Apoplastic and symplastic pathway. Tell me, guys. Samaira, Minashi, Atif, Alvia, tell me. Apoplastic, symplastic pathway clear?
Yes, sir.
So start coding now. First, put the heading apoplastic pathway. Here, water passes from the root to xylem. Here, water passes from root to xylem through the wall of intervening cell without crossing the wall of intervening cell, without crossing any membrane or cytoplasm, without crossing any membrane or cytoplasm. Full stop. The pathway provide least resistance. The pathway provide least resistance to movement of water. The pathway provide least resistance to movement of water. Let's stop. This is called apoplastic pathway. Let's stop. This pathway in the same paragraph, this pathway is interrupted at endodermis, which have this pathway is interrupted at endodermis, which have Casparian strip, which have Casparian strip. Which have Casparian strip. Full stop. Next heading. Symplastic pathway. Next heading. Symplastic pathway. Water passes from cell to cell. Water passes from cell to cell through. Water passes from cell to cell through their protoplasm. Full stop. It does not enter cell vacuole. It does not enter to cell vacuole. Full stop. The cytoplasm of adjacent cells. The cytoplasm of adjacent cells remains connected. The cytoplasm of adjacent cells
It
remains
does not
connected.
enter.
Sorry,
Can you say from it does not enter?
it does not enter into central vacuole or cell vacuole. It does not enter into cell vacuole. Full stop. The cytoplasm of adjacent cell are connected through the cytoplasm of adjacent cells are connected through plasmodesmata. This one, plasmodesmata. Cytoplasm of adjacent cells is connected through plasmodesmata. Full stop. For entering into symplast, water have to cross plasma membrane. Let's stop. Is it clear?
Yes, sir.
Put a note below this. Put a note. Mineral nutrient also have the same pathway. Mineral nutrient also have the same pathway. That of water. Mineral nutrient also have the same pathway that of water. Right? So this is the apoplastic and symplastic pathway. Now, one more thing. What is the mycorrhiza? Can anybody tell me what is the mycorrhiza? I think we have studied it. Yes, Samira? Did we discuss it, mycorrhiza, in fungi? Yes or not? I think I have discussed that. Myco, double R, H, Z, I, Z, mycorrhiza. Look, mycorrhiza is a symbiotic relationship. Basically, in case of gymnosperm, right? The roots of gymnosperm are not that much stronger. So gymnosperm is a kind of higher plant. Gymnosperm. So gymnosperm and angiosperm both belongs to both are higher plants. The demerit with gymnosperm that their roots are not that much stronger, say that they can withdraw water from anywhere. Angiosperms have very strong roots, but not in the case of gymnosperm. So what is that? Around the root of gymnosperm, there is a covering of a network of filament. They are fungal filament, right? So these are the fungal filament, right? So this is the fungi, and the above part, that is the root part. This is the root. So what happened? Fungi give water and mineral to root. Water plus mineral. In return, root gives fungi food, right? So both, what kind of the relationship? Both have symbiotic relationship. Symbiotic relationship. So symbiotic, both are helping each other, right? So the symbiotic relationship between the fungi and roots of higher plant is called mycorrhiza. Is it called mycorrhiza? Is it clear or not?
Yes.
Yes.
Example, what is the example? Root of orchid, pinus. Please note it down. Mycorrhiza is a fungal association with roots of higher plant. Mycorrhiza is a fungal association with root of higher plant. Full stop. Hypha are specialized to absorb water. Hypha are specialized to absorb water. Full stop. And in return, root gives, food to fungi. Full stop. This symbiosis is called mycorrhiza. This symbiosis is called mycorrhiza. Right? Is it clear?
Yes.
Okay, great. Now the next part is, that is the, yes, transpiration of water. First, we will study the transpiration of water. No, not transpiration of the water. There is a root pressure theory, then guttation. Right. Let me tell you what is the guttation, what is the transpiration. Two things: transpiration and guttation. Just a minute. Give me one second. Look, there is a difference in guttation and transpiration, but both are the important for you. Just a minute. Mhm. Transpiration is the you are just studying since your childhood. What is a transpiration? So you must have listened even in you must have studied even in your class 10 transpiration process. And gradation is something different process. So next one is the next heading. In fact, okay, the next heading is transpiration, and then we will study gradation. So this is the transpiration process. So in transpiration process, there is nothing very extraordinary, but still I want to add on something. So where the maximum number of stomata are formed. So look, transpiration is just water expansive process. Transpiration always give loss to the water to the plant. Plant earn water with very hard. So the upper surface that is called adaxial surface. Adaxial surface. It have low stomata count, sometimes zero stomata, to reduce transpiration. While the lower surface, that is called abaxial surface. Abaxial surface that have higher stomata count. Higher stomata count. It have higher stomata count. That is, so it have more number of stomata, right? So transpiration takes place more from the lower side, right? Please note it down. The loss of water from the exposed part of plant called transpiration. The loss of water from the exposed part of plant is called transpiration. Full stop. (98 to 99% of water, 98 to 99% of water absorbed by plant is lost in transpiration. Is lost in transpiration. Full stop. Hardly 0.2%, hardly 0.2% is used in photosynthesis, while the rest is retained. While the rest is retained. While rest is retained in the plant for growth. So 99%, 98 to 99%, that is in transpiration. Transpiration. While 0.2%, that is utilized in photosynthesis. Photosynthesis. And rest 0.8%, that is utilized in plant growth. So that is the calculation of water, right? And the rest we will study. Like a very small part of the chapter has been left. That is a transpiration. From transpiration, we have to study the two theories only: mass flow theory and the transpiration flow theory. And then we'll complete this chapter. In next class, we'll complete the chapter and move on the next chapter. Clear? Is it clear? All clear or not? Samaira, Minashi, please tell me. Alvia.
Yes, sir.
Okay, fine. So see you in next class and we'll continue with it, right?
Yes.
Bye bye. Take care. Good night, all of you.
Thank you, sir.
Welcome, welcome, beta,
Thank you, sir.
welcome. Bye. Welcome. Good night.